Okay, we're looking at Benchling and why it's such a useful tool to use for the rest of the work that we're going to do for molecular cloning and modern biotech. So you're going to start at the Benchling site where you sign in or create a new account. And once you do that, to take it into your account, and there's some things that you need to just pay attention to down here. So if you look up here, this is where you start, and it's got the date and the different things that you've got to do. So it's ideal for planning your work. So you can see that there's a calendar here, and depending on the things that I have put into my Benchling, into my projects, into my folders, it will come automatically here in the calendar, which is really cool if you're trying to organize your work, especially if you're doing a project or you're trying to organize your assignments and so on. So by clicking that, you'll be able to see at a glance what you're supposed to be doing, and you can also color code things according to project, so you can see which project comes when and so on, and be organized in your work. And if you come here, this is where you can start new projects. So I put my different activities here, and this is your activity here for the onboarding. And I haven't done much here, but I'm just showing you where you'll find most of the material that comes after this that you'll use to work through the assignment I gave you. Then if you come down here, you can create an entry, a protocol, or sequence, or different types of things. So there's two versions of this Benchling. There's Benchling, which is the paid version, where the institution, such as CBU, will be paying for the subscription, which comes up to like $15,000 to $20,000 per year. And that would allow the scientists to be able to work with a full package. And so when you click here, there'll be a whole bunch of different things here. But this one is the free version. That's why when you click here, you don't see anything. And there's some features which are not there, but you don't need them. So all you need is to be able to use the projects, the entries, protocols, and these things here. So what we'll be doing for your homework, which was basically asking you to create a new sequence of GFP and also to prepare a primer. So how do you use this thing? You have to remember that there's two sections as well. There's the molecular biology section and the notebook section. The molecular biology section is the section you go to when you're trying to do your cloning, make your constructs and all that. Your notebook is the section you go to when you are making your actual journal. So instead of having a physical lab journal, you'll be using this as your journal. So if you explore the notebook, it has a collection of all the entries that you've made in there. So these are the entries I've made. And the easiest way to look at it is like a journal, where you have pages, and every time you want to put in a new page, you say create entry. So you can create a blank entry like that. You name it entry 1, and you put it in a project that you've already made. So I've put example project here. And again, projects are made up here. That's how you organize your whole work. So here's entry 1, and this is what it looks like. So you can imagine this as your lab journal, and it keeps tabs of everything that you're doing. So it keeps the date, and then it allows you to enter the different things that you're going to do. But it also helps you to plan. So if I'm trying to do this assignment 1, right? So the GFP assignment. Of course, you'd give it a better name than that. What do I have to do? I can consult my the actual homework, and it said something like collect the sequence from NCBI. Then I had to import it into Benchling, and it needed to be circular, I think. Visualization. And then there was the primer story as well. So the primer design. So this is just giving you an idea of how you can organize what you're supposed to do. So you say, okay, I'm going to do these things. And then I start with the first thing, which is the sequence from, so GFP sequence retrieval. GFP sequence retrieval. So this is the first thing I'm trying to do. And I'm going to give it a nice header. So this is my work, right? And what am I doing? If I get it from NCBI, in the directions, I was told to look for this, wasn't it? This Aquaria Victoria green fluorescent protein mRNA. There was an accession number that was given in the homework. So all I did was put it in here, and this is what came out. So if I look at the accession, I've got the protein sequence here. I've got the gene here. And so I can import this, and it said in the instructions that I need to send this to file, and then I needed to download not the faster version but the GenBank version. So then I do that, and then it will download to the place of my choice. So I'll say GFP sequence 1. Okay. This was for our own purposes. Okay, and I've saved it in this folder. Save. So then it told me in the instructions that I needed to import it by making an entry and then just importing it without opening it first. So here I could make another entry, or it said import it as a sequence, isn't it? So I import it as a DNA sequence. Where am I getting it from? I could have just put in the accession number right here because this Benchling is linked to NCBI, but I've already downloaded it, so it's a DNA file. I'm going to choose that file which I just downloaded, and it was called, what was it called? What was it called? This one. You see, it's GFP sequence 1, and it's a GenBank file. So I open that, and it's uploaded it. Then here, I'm going to ask it to do all this stuff. There's also somewhere where it asks you whether you want it to be a linear or circular file. So this particular one is linear because it's just the gene. It's not in a plasmid or a vector. Right, so let me close that, and here it is. So this is my sequence right here, AEVP GFP, and it tells me how many base pairs it is, and there's all these amazing things that you can see also. If you look carefully in the way it's displayed, and you can choose different display things up here, annotations, primers, cut sites for restriction sites, amino acids, and so on. We'll learn more about those as we go. So it tells me even where the start codon is. Remember that start codon is always methionine. And so there's GFP, and this is the codon sequence. And I've got my DNA here. Then I've got my amino acid sequence underneath. So you can see that for every amino acid, there's 3 DNA bases, just like that. And over here, I've got exactly where I'm looking at in the sequence, and all the annotations are here. So it can also help me if I want to see different open reading frames. So it will show me the different open reading frames 1, 2, and 3. Or I can say no, don't show me all that stuff, which is what it's done. Now it's only 1 reading frame. And my whole protein sequence is there all the way. And one of your questions was, where's the start and the stop codon? So you can actually look and find them in these annotations right here. So you can make this a bit bigger so you can see. Yeah. All the way. Yeah. So this is all that you're trying to do. And what I want you to be able to do is see the DNA sequence together with the protein sequence so that you can make sense of the actual protein, and then be able to look through for stop codons. So I haven't mentioned them. I want you to scan this sequence to find where the possible stop codons are. And then you can also add new annotations. So here where it says create, you can create a new annotation, meaning if you want to put in something like a stop codon or a restriction enzyme site, you can do that using this, and we'll learn how to do all that. So for now, if I'm finished with this, I would download that, create PDF there, and then it has a PDF of my sequence. And now that PDF is free for me to, I can use it to actually put back into my work. So if I go back to my project, and where's my example project, the one I was working on, the GFP assignment. So here we are, GFP sequence retrieval. I can also get the FASTA from here and copy paste that like this. So that is my FASTA right there. Then I can also look at the PDF that I just made and also put it in here. You can insert all sorts of things like attachments. So I could actually attach that PDF that I made and basically put all sorts of things in my homework as I've done it. So here I've done this first part, I've retrieved the sequence, I've collected it, and I can tick that. Then I imported it into Benchling, I can tick that. Then for the circular visualization, if it was in a plasmid, I'd be able to do that, but the one that I have is not. I'll show you one, which is a circular one. So if you go to this resource called Add Gene, you can actually get plasmids which already have things inside. So if we look at this plasmid, which I was looking for, I went to Add Gene, and then here in the catalog, I looked for GFP plasmids which have GFP, and I found this one. So this one is called eGFP PBAD, and here's the GFP. This gene actually expresses GFP, and because it's a plasmid, there's ampicillin resistance, there's the origin site, and all the rest of it. So this is an example of a plasmid, and if I actually imported this sequence from here, I could, when I import it, make it a circular version and have it like that. So I can actually visualize this whole thing. I can visualize this inside my Benchling. So if I wanted to be showing off a little bit, I can say GFP in a plasmid, just to show what it would look like. And then I can either let's see, I can download this sequence and import it into my Benchling, or I can download this very PDF. So here it says I can download the GenBank file. So there we are. Okay, and we do the same thing again. Add sequence, new sequence, upload file, DNA. Where is it? It was called eGFP. I think it was this one, empty backbone. I may be wrong. So there it is. Let's get it all with the annotations and everything. Did I give it a name? I don't think I did. It will probably keep the same one. So now here it is. beautifully done, where you can see all the things that are interesting. Again, you can create a PDF of that, or you can just take the sequence, or there's a whole bunch of things you can do, but the same story is here, where you can see the sequence up here, and then you can see the different annotations. So you can see where the different genes are, the different markers are. You can click here and it will show you where that is on the actual sequence. And the primary sequence is there for the protein as well, which is super useful. And then you have this star. What does the star mean? It means a stop codon, right? So we had the ATG as the start codon. And if we look at this gene, this is RRC, and it's starting here. If you look at the arrow, it starts here and it goes that way. That's why the stop codon is at the end, and the start codon is at the is further down, so it's backwards. So here's the methionine that we saw. Remember the methionine we saw in the other one? There's RSC gene, so it actually starts here and it goes this way. And you can see from the arrows in the amino acids that it's going this way. So that's basically it. If we look here, this is the GFP now. There it is. It's annotated, EGFP. So this is the region that's actually showing you the part that's encoding the protein. So this plasmid is for inserting your own gene before, and then it's fused onto the GFP, so that when the GFP and your gene are expressed, your protein is expressed with GFP attached to it, and GFP shines, and then it will tell you where your protein is located in the cell. And there's other purposes as well for making these fusions. We'll talk more about that later. So at this point, all you have to do is be able to bring in your sequence like this and use it to make new entries in your notebook. So if I wanted, I could copy that. Which bit? I could copy the DNA sequence or the complement, or the amino acid itself, and that all is of my GFP. So if I went to my GFP assignment and I say GFP in a plasmid, and in here I would insert whichever the document that I made, the PDF or whatever. So I could do that later, but here I want to put GFP protein sequence that I just got. There it is. So you can basically compile your whole homework in this tool. You can do your analysis using the molecular biology tools, and then you can note it down using the notebook tool. And then it has a date stamp which tells me exactly when you did this. And then for primer design, we'll talk more about that later, but I'd love you to investigate it, because you can just look here and look at these, analyze. It says about primers and so on. Do you want to create a primer, a forward primer, a reverse primer? So try that, see what you can get. But this is the tool we'll be using for all the work that we'll be doing in future. And that's it. So when you're done, you should have an entry in your notebook like this. And then what I do is I go over here and I can see all the people who have signed up. There you are. And I can pick on one of you and check out the things that you have access to. So I'll give you access to like this same one, and then I'll be able to view the things that you've been able to do. So that's how we share the work. And this becomes useful also later on when I want you to have access to protocols or different things, different analyses of the proteins we're working with, and a lot of different things. And this is another function which is using AI generation to generate 3D structure from your sequence data. So this thing has been running over the weekend because it takes quite a while. I put in this like 2 days ago. I just put in the sequence and I asked it to do the 3D structure based on the GFP sequence that I gave it. And this is the sequence that it gave me. And this, again, we'll talk more about it. But these are the different aspects of this tool, and that's what I'd love you to be able to be free to use. So have a go. We'll discuss it in class on Thursday.